All right, neurotransmitters and neuromodulators. So these guys are both chemicals that can convey information among neurons. Neurotransmitter is released by a presynaptic neuron and acts directly on a postsynaptic ion channel or activates proteins inside the postsynaptic neuron. So what we mostly have been talking about up until this point is neurotransmitters. Neuromodulators are released into the extracellular fluid and adjust the activity of lots of neurons, not just one. So the same chemicals, the same neurotransmitters can act as a neurotransmitter or a neuromodulator. So the difference between the two is that neurotransmitters affect the postsynaptic neuron either directly by activating an ion channel or indirectly by activating proteins inside the postsynaptic neuron. Neuromodulators adjust the activity of many neurons. There's also a difference in the speed of how they act. So with neurotransmitters, there are two types of neurotransmitters, and they can be either excitatory or inhibitory to the postsynaptic membrane. So the two types of them are fast acting, they act directly, and transmission requires less than 1/1000th of a second, so super fast. Slow acting, the ones that act indirectly, transmission requires about 1/10 of a second to minutes. So a little bit slower indirect action. And the fast acting, they have a short-lived action. The slow acting have a little bit longer life. So they regulate fast-acting neurotransmitters by influencing the amount of neurotransmitters at the pre and postsynaptic membranes. So depending on what they bind to, so I'm going to just say this right now, and I'm going to say it again later, and I'll say it a lot. The effect of a neurotransmitter depends on the receptor it binds to. So you can have the exact same neurotransmitter bind to a different receptor and have a different effect. So sometimes they can be inhibitory or excitatory depending on what receptor they bind to. Neuromodulators alter neural function by acting at a distance away from the synaptic cleft. So neurotransmitters, they work at the synaptic cleft. Neuromodulators are at a distance from the synaptic cleft. The effects manifest more slowly and usually last longer than those of neurotransmitters. So the neurotransmitters happens in seconds, and the effects last for neuromodulator happens in seconds, and the effects last from minutes to days. We know that the neurotransmitters act either very short, like 1/1000th of a second to 1/10 of a second, and the effects last for minutes. So neuromodulators are slower acting, but they have a longer effect. Neurotransmitter agonists and antagonists are drugs that affect the job of neurotransmitters, basically. So a lot of the like the medications, like serotonin reuptake inhibitors, which is Prozac and those sort of things, Prozac, Zoloft, that sort of thing. They are agonists. There are some things that prevent binding, and those are antagonists, like Chantix, the anti-smoking medication, that is a neurotransmitter antagonist. And nicotine can be a neurotransmitter. It's a nicotine antagonist. So agonists are drugs that bind to the receptor and mimic the effects of naturally occurring neurotransmitters. So carbidopa, levodopa, that's used in treatment for Parkinson's, its trade name is Sinemet, It is a dopamine agonist. It mimics the effect of naturally occurring dopamine, because dopamine is the neurotransmitter that's missing for the most part in Parkinson's. Antagonists are drugs that prevent the release of neurotransmitters or bind to the receptor and impede the effects of a naturally occurring transmitter. So Chantix, that's an antagonist. It's a nicotine antagonist. You can have drugs that affect the neurotransmitter action in different ways. So when we start talking about specific neurotranslators and neuromodulators, there's a lot of really detailed chemical information in the book, which is great, but we don't really need to know it to that level. Basically, I just want you to know the ones that I have listed here and their effects, whether they're inhibitory or excitatory on the postsynaptic membrane. So there's a nice table in the book, table 6.1, talks about categories of amino acids, the different neurotransmitter and neuromodulator, and whether they're excitatory or inhibitory. So that is a lovely chart to study from. So just talking about some specific ones, there's also a really nice Venn diagram in the book. I love Venn diagrams. And it talks about the comparing the effects of serotonin, norepinephrine, and dopamine. And they all have effects on emotion, cognition, and mood. But then they have other effects. So it's a great Venn diagram. Take a look at that when you get a chance. So acetylcholine is the major conveyor of information in the peripheral nervous system. It has slow-acting effects in the PNS that regulate heart rate and other autonomic functions. Receptors that bind acetylcholine fall into two categories, nicotinic and muscarinic. So in general, acetylcholine is excitatory. And it is the neurotransmitter that functions at the neuromuscular junction. And of course, when you're getting a muscle impulse, you want it to be excitatory. You want the muscle to contract. You're not going to send an inhibitory impulse to a muscle. So acetylcholine, and then it's a big player in the autonomic nervous system, and we'll talk about that a little bit more in the autonomic chapter. So the next group, so the acetylcholine is in a category called cholinergic, and the next category is the amino acids group. So glutamate, glycine, and GABA are the three in that group. There are some other ones listed in the book, but these are the ones we're going to talk about. Glutamate is principally a fast excitatory transmitter of the central nervous system. It's sometimes overactivity of glutamate with NMDA receptors can cause epileptic seizures. So there's also a real big function of glutamate in the cell death cascade after stroke or an injury, and we'll talk a little bit about that in the neuroplasticity chapter. Glycine inhibits postsynaptic membranes, particularly in brainstem and spinal cord. And GABA activates slow-acting responses. It's linked to ion channels and second messenger systems. So really just know that basically GABA is inhibitory and glutamine is excitatory and glycine is inhibitory. So that's about the level of information that I want you to know. So where it functions and whether it's excitatory or inhibitory. So the next group is the amines, and serotonin, dopamine, and norepinephrine are part of the amines group. And those guys are really important on our mood and motivation. So those guys work together, the amines work together on a lot of those things. Dopamine can be excitatory or inhibitory depending on the receptor. It affects motor activity, cognition, and behavior. So the Parkinson's is a lack of dopamine because dopaminergic cells are dying off, and it's categorized as a motor dysfunction, but it also has a big effect on cognition and behavior. So because dopamine is a big thing in that, motivation and cognition and some sensory effects as well. So because it has different effects depending on where it binds, it has wide-ranging effects when you don't have enough of it. So norepinephrine plays a vital role in active surveillance by increasing attention to sensory information. Norepinephrine is a major player in the sympathetic nervous system and its response to what happens, because if we have a sympathetic response, immediately our attention to sensory information goes up. That's really important for avoiding threats, basically. Serotonin, norepinephrine, again, just like dopamine, can be inhibitory or excitatory depending on the receptor. Serotonin affects sleep, general arousal level, cognition, perception, motor activity, and mood. It's usually inhibitory. So obviously those things are all associated with depression, or can be. Antidepressants are often serotonin reuptake inhibitors, so the serotonin stays longer in the synaptic cleft. Histamine is another one of the amine neurotransmitter neuromodulators, and it's concentrated in the hypothalamus. Histamine, has a lot of effect in the inflammatory response in the body, so it has to do with how we respond to things. And histamine is usually inhibitory. Interesting. So the peptides, the opioid-peptides, which are the endorphins, enkephalins, and dynorphins, those are endogenous opioids that inhibit neurons in the central nervous system, and they're involved in the perception of pain, altering the perception of pain. So the other peptides are also involved in pain. Substance P, which its name kind of gives you, like, it stimulates nerve endings at the site of injury, so it is excitatory, increasing pain response. Calcitonin, which is a gene-related peptide, acts as a neuromodulator. So nitric oxide, it's in its own category because it's a gas. It regulates the vascular system in the periphery and is also active in the brain. It doesn't require a receptor on the outer cell membrane for activation, and it's involved in persistent changes in the postsynaptic response to repeated stimuli and in cell death of neurons. So that one is acting more as a neuromodulator than a neurotransmitter. It's usually excitatory.